Hi, everyone. Thanks for joining us for this webinar today. It's going to be focused on Benchtop long-read sequencing on Vega, faster and cheaper with SPRQ-Nx. So we're joined today by many great speakers. Greg Young is going to be speaking, but is not going to be present for the live Q&A. Instead, we're going to have Matt Boitano, which is the platform product manager, also joined by Nadia, Gianluca, and myself, going to be giving presentations throughout the portion of the webinar. So we do have a lot of great material for you to cover today, and the presentations portion of this webinar is pre-recorded, but it is followed by a live question and answer session. You are welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee control panel, and we'll get to them either in the chat or at the end. Just know that all attendees are muted. We've also uploaded a few pieces of literature to the control panel, so please feel free to download those. We will be recording this webinar and making it available in the next couple of weeks, so please keep an eye out for a follow-up email on that. And then immediately following this webinar, you'll receive a brief questionnaire. Please do take the time to fill it out as it helps us understand your needs and plan for future webinars. If you do encounter any audio issues as we switch between the live conversation and the pre-recorded talks in the webinar, just make sure you have the right audio mode selected. So please check the audio tab on your control panel to make sure you have selected that correct audio output depending on the device you're using. It also may help for you to leave and rejoin the webinar if you still can't hear. And with that, we're going to go ahead and get started.
Hi, everybody. My name is Greg. I'm a senior product manager at PacBio, and today I'm going to talk about benchtop long-read sequencing on the Vega system. So PacBio HiFi sequencing provides a more complete view of biology than any other single technology approach. HiFi enables T2T genomes. It detects small and large genetic variation across more of the genome. It produces high-quality reference assemblies of many challenging plant and animal genomes, and adds functional layers of information with native methylation detection and full-length isoform sequencing. All of this is possible because of the improvements in the technology over the last decade. In that time, we've seen approximately a 10-fold increase in sequencing throughput. Some of the highlights include the introduction of SPRQ chemistry on the Revio system back in late 2024. That update provided a 4-fold reduction in DNA input, which enabled sequencing using just 500 nanograms of genomic DNA. SPRQ chemistry also provided increases in sequencing throughput and multiomic capabilities. The latest updates on Revio, launched back in May, brought smart cell multi-use and expanded the multiomic capabilities to 5-hydroxymethyl-C detection. With these latest updates, the cost of doing a long-read human genome has dropped to just $345 using the Revio system with smart cell multi-use. Now, I will point out that all pricing showed here is in US dollars at list price. But we're not done this year with innovations. We're going to bring the SPRQ-Nx chemistry from Revio onto the Vega system later this quarter. Now, Vega has already removed barriers for labs wanting to get into long-read HiFi sequencing because of its relative low cost at $169,000 US. The SPRQ-Nx chemistry on Vega is going to remove some of those additional barriers. For example, one historic barrier for long-read sequencing has been the DNA input requirement. SPRQ-Nx chemistry will reduce the single sample DNA input to 500 nanograms when working with a PCR-free workflow. But let's not forget, we do have the Amplify protocol that enables DNA inputs as low as a single nanogram when doing PCR. I'll get into more of the details of SPRQ-Nx on Vega in a moment. But first, I wanted to give a brief background on HiFi sequencing for those in the audience that are new to PacBio or long-read sequencing. So this next section is going to be a brief crash course in HiFi sequencing. PacBio sequences single molecules of DNA, not amplify clusters like standard NGS short-read systems. Prepared fragments of DNA are isolated in tiny nanowells on the surface of what we call the SMRT cell. Our sequencing polymerase is a strand displacing polymerase. This enables the polymerase to sequence both the forward and reverse strands of the same DNA molecule. The base incorporation is captured by the imaging of fluorescent phospholink nucleotide analogs. And because this is all monitored in real time, we can use the polymerase kinetics, mostly the time between base incorporations, to infer positions of DNA methylation. Over the years, protein engineering and sequencing chemistry innovations improved the lifespan of the sequencing polymerase. This has enabled the polymerase to make multiple passes on the forward and reverse strands of the DNA because we circularize the DNA with hairpin adapters. Now, each pass on the forward and reverse strand is collected as a subread, and those subreads are aligned, the adapter sequences are trimmed, and a consensus sequence is called. It is this consensus sequence we call the HiFi read. It has an average of 99.9% accuracy, or Q30. For those of you that may not be familiar with the Phred scale, that is one error in 1,000 bases. The DNA fragment lengths in HiFi sequencing can range from anywhere down to 500 base pairs, upwards of 30,000 base pairs or more. But the average is generally between 10 and 20 kilobases for most whole genome sequencing applications. The multiple passes on the DNA fragment not only generate an accurate sequencing read, but also enable accurate methylation calling. The technology measures both the time it takes to incorporate a base and the time between base incorporations. So at sites of DNA methylation, the polymerase will slow down, increasing the distance between signals. We call this the interpulse distance, and it's used to infer sites of DNA methylation. Each DNA modification will have its own unique kinetic signature. So from a single data type, you can get both a highly accurate DNA sequence and methylation calls. PacBio HiFi reads combine the best of both worlds. That high accuracy you are used to with short reads with the length and native methylation of long reads. This enables haplotype phasing across genes and long genomic distances. It also enables the mapping of sequences to more complex regions of the genome that are often medically or biologically interesting. An additional differentiator in the long read space is that PacBio HiFi has integrated on-instrument primary analysis and demultiplexing with additional secondary tools in SmartLink, simplifying users' workflows. A bit more on HiFi as a data type. HiFi reads are long reads. For whole genome sequencing, the recommended insert size or read length is generally around 15 to 20 kilobases. But as I mentioned previously, the reads can range anywhere between 500 bases upwards of 30 kilobases or more. This means you can directly span variants or repeats, or for other applications, you can get full-length amplicons or full-length messenger RNA for isoform characterization. And as I've already highlighted, methylation calls are provided as well, requiring no additional chemistry or assays. This unique combination means you can get haplotype resolved variants and methylation from a single data type and experiment, making it a cost-effective choice over alternatives requiring multiple assays and/or sequencing runs. In addition, the accuracy and read length allows for confident mapping in repetitive regions of the genome, helping resolve tandem repeats in medically important regions of the genome. PacBio has a broad and multi-omic set of applications that can be run on both the Revio and Vega systems. This includes applications like de novo genome assembly, small and large variant detection, either at the germline or somatic level, epigenetics with our methylation calling, targeted resequencing using either panels or discrete amplicons. We have the pure target application for PCR-free targeted sequencing that can be used to detect and characterize repeat expansions. We have the sequencing of AAV constructs and the shotgun sequencing of microbial isolates and metagenomic samples. On the RNA side, our Kinnex applications enable the full-length sequencing of messenger RNAs at either the bulk or single-cell level. Or we have the targeted 16S ribosomal RNA sequencing at scale. All right, here's an example that highlights both the power and cost effectiveness of HiFi reads. So the ability to detect 6-methyladenine modifications enables the Fiber-seq assay, which adds chromatin structure to the underlying sequencing reads. That means from the same assay, on the same sequencing read, you can get multiple dimensions of data, right? You can get the variants, so you can phase each of the alleles in a haplotype-specific manner. You can look at the methylation, the 5-methyl-C and 5-hydroxymethyl-C on a per haplotype level. And then with that added Fiber-seq information, you can see the chromatin accessibility or nucleosome dynamics on a haplotype-specific level. Right. For many labs, HiFi sequencing has been something they've accessed only through a service provider. SPRQ-Nx on Vega is going to remove some of those additional barriers and better enable you to bring HiFi sequencing into your own lab on your own schedule. So SPRQ-Nx chemistry is going to expand the flexibility, accessibility, and utility of the Vega system. It brings a 50% increase in HiFi yield, helping make long-read sequencing more affordable with a 40% savings in the cost per gigabase. A 4-fold reduction in DNA input makes more samples accessible to long-read sequencing, reducing concerns around sample qualification and applications. And it brings increased multiomic capabilities with the addition of 5-hydroxymethylcytosine calling, as well as improved 5-methyl-C and 6-methyladenine calling. There are also platform-specific updates. We're introducing shorter runtimes of 2 and 4 hours for small insert libraries such as amplicons. And this is going to enable same-day sequencing results. So now with SPRQ-Nx chemistry on Vega, you'll have the option to do 2, 4, 12, and 24-hour runs. We're also introducing 21 CFR Part 11 compliance support for things such as system sign-in to both the instrument and SmartLink software. With those shorter runtimes, you can get same-day sequencing results for short amplicons. So for a 1 KB amplicon, you can get over 6 million HiFi reads with 95% or more of the bases at Q30 or higher, with a total time to results of 5 hours. So we have a 1.5-hour sequencing prep time on the instrument, a 2-hour sequencing time, and then a 1.5-hour primary analysis time. For larger 2 KB amplicons, the time to results increases to approximately 8 hours with the longer sequencing run. This means for sequencing, 1 KB amplicons, you can start in the preloading feature on Vega. You can organize your runs back to back and get, information or sequencing experiments, 2 sequencing experiments in as little as 8.5 hours. Here's another example of what's possible on Vega with SPRQ-Nx. With the Fiber-seq assay and a single 24 sequencing acquisition, you can generate whole genome sequencing data at 30x coverage. You can get genome-wide methylation, plus chromatin accessibility and nucleosome positioning data for under $1,000 in sequencing consumables. Also new in SmartLink is the ability to look at methylation distributions on a per read basis. And this will make it easier to QC your Fiber-seq experiments without having to run the FiberTools application. Moving forward, the Vega and Revio systems are going to share the same sequencing chemistry, but they are different systems for different needs. The Vega system is going to be that low cost entry point system for individual or small core labs that want to do, maybe 100 to a couple hundred samples a year. Whereas Revio is a much higher throughput system, and it can run multiple SMRT cells at a time at a lower per run cost. It's going to be a good fit for those labs needing to sequence hundreds to thousands of samples per year. Even though both systems are going to share the benefits of the SPRQ chemistry that I just mentioned, like the lower DNA input amount, It's only going to be the Revio system that's going to have the ability to take advantage of the multi-use SMRT cells. So that's going to be one big differentiator between the two systems. Now, in the next section of this webinar, we're going to have a panel of speakers that are going to talk more about what the Vega system can do with the SPRQ-Nx chemistry, now that you have a good understanding of the new features that the SPRQ-Nx update is bringing to the Vega system. We're going to talk about targeted resequencing applications, right? And this involves both amplicon sequencing for clinically relevant variants. It also includes our PCR-free pure target approach that's used to really characterize and detect repeat expansions. We're also going to talk briefly about AAV sequencing and how it can be used as a quality control, and full-length RNA sequencing of both isoforms and vaccines. We're also getting into doing microbial and metagenomic sequencing. This includes 16S ribosomal RNA, amplicon sequencing, metagenomic shotgun sequencing, and microbial isolate sequencing.
Hello, and thank you for joining. Today, I will focus on how Vega, together with the new SPRQ-Nx chemistry, expands target HiFi sequencing. For many PacBio customers, HiFi already means long, accurate reads, phasing, structure variant detection, and better resolution of difficult regions. Vega and SPRQ-Nx brings those strengths to more focused, targeted resequencing workflows. For customers using Illumina or other short-read platforms, Vega can also be a practical entry point into long-read sequencing where short reads leave uncertainty. The question to keep in mind is which high-value target in your lab would be easier if you could read the whole molecule with high accuracy. Targeted sequencing remains important because it's practical. It focuses sequencing, depth, analysis, and cost on the genes or regions that matters most. But targeted assays range from routine single genes to larger panels and complex rare disease targets. As the biology gets harder, the need for long, accurate reads becomes clear. This slide summarizes the Vega and SPRQ-Nx value proposition: higher confidence for difficult regions, broader flexibility to consolidate targeted assays on one platform, and clinical research readiness for controlled environments. The goal is not just more data, it's more complete, more interpretable data from the targets your lab already cares about. Now, this slide lists genes many labs already know well: HBA1, SMN1, F8, HLA genes, and others. These are not obscure targets. Many are already on test menus and in research workflows. They are difficult because of repeats, high homology, pseudogene, structure variation, copy number changes, or the need to phase variants across a region. So the customer question is not simply, can long-read sequence these genes? The better question is which difficult genes or target in your current workflow could be simplified, consolidated, or made more informative with target HiFi sequencing. Targeted HiFi sequencing is not limited to one application area. Across PacBio, targeted approaches are used in human genetics, microbial metagenomics research, which you will hear from Jeremy later in the webinar, and biopharma application, which you will hear from Nadia, and other areas. For this section, I will stay focused on medically relevant targeted resequencing applications. The broader point is that the same principle applies across applications. Enrich the targets that matters, and then use long, accurate HiFi reads to get more complete views of that target. Now, there are several ways to enrich targets for HiFi sequencing, and the best choice depends on your specific goal and needs. Amplicons are excellent for small panels, focused genes, high-throughput workflows where cost per sample and speed matter. PCR-free pure target is valuable when PCR is part of the problem, when repeats, methylation, and extreme GC or very complex regions needs to be preserved. Hybrid capture is a flexible path for medium to large panels and is familiar to many sequencing teams. Different enrichment methods, same outcome: focused sequencing with long, accurate HiFi reads. The amplicon message is straightforward. If you can amplify it, we can sequence it full length. With short-read NGS, a long regions often becomes a talent exercise with multiple amplicons and short paired-in reads. With Sanger, a long-range PCR product may still require primer walking. Every additional primer pair adds design work, optimization, and possible failure modes. HiFi amplicon sequencing changes that experience. One long molecule, one full-length read, and the ability to see variants together in phase across the target. For short-read customers, this is often the easiest way to see long-read value proposition. Instead of reconstructing the targets from short pieces, you read across it directly. This is where Vega and SPRQ-Nx become very practical. For targeted amplicon workflows, the runtime can be short enough to support a sample to report in less than 24 hours, depending on the assay design, batching, and analysis. The example shown is a thalassemia amplicon workflow with up to 2.5 kilobase targets and a 4-hour sequencing run. The message here is that targeted HiFi sequencing can resolve difficult biology and still be operationally practical for focused assays. In pharmacogenomics, CYP2D6 is a strong example where long, accurate reads matter. This region is challenging because CYP2D6 sits near the CYP2D7 pseudogene. Sequence similarities can create mapping ambiguity, coverage gaps, and difficulties assigning variants to the correct gene or haplotype. In the PacBio HiFi amplicon example, CYP2D6 amplicon is captured specifically and sequenced across the region. That enables full phasing across more than 40 heterozygous variants and avoids misalignment to CYP2D7. This is the kind of target where long reads can directly improve interpretability. Amplicons are powerful for focused targets, but many labs also need larger panels. This is where hybrid capture becomes important. The PacBio compatible partner products shown here differ in content, input requirements, and panel design, but the overall concept is similar. Prepare index libraries, capture target regions with probes, amplify after capture, convert to HiFi libraries, and sequence using long, accurate reads. For PacBio customers, this extends HiFi into larger targeted panels. For Illumina customers, this is an approachable bridge because targeted capture is familiar, but the readout can be more informative for difficult regions. This GPA example shows why hybrid capture with HiFi reads can be valuable for difficult genes. Here, we compare short-read whole genome data with HiFi targeted enrichment data for HG001. In the HiFi targeted enrichment data, the GBA region is covered across the gene and phased into haplotypes. In the short-read data shown here, there's a coverage gap and less uniform coverage. The key is that long, accurate reads preserve context across the gene. That context can reduce uncertainty, support phasing, and help avoid follow-up testing or sendouts when a targeted HiFi assay provides a more comprehensive view. Now let us move to Pure Target. Pure Target is designed for cases where amplification can get in the way of the answer. It is a PCR-free, CRISPR-Cas9-based enrichment method for challenging targets such as repeat expansions, extreme GC-rich regions, and applications where native DNA information, including methylation, is important. The workflow starts with high-quality, high molecular weight DNA, guide RNA, direct Cas9 cutting around the targets, smart bell adapters are ligated, non-target templates are removed, and enriched molecules are sequenced on Vega or Revio. The customer hook is powerful. One targeted HiFi workflow can connect sequence, structure, repeat size, and methylation context in the same experiment. This slide shows the consolidation opportunity for repeat expansion detection in ataxia. Historically, labs may use several methods: PCR and capillary electrophoresis to identify expansions, repeat primer PCR to size expansions, and in certain cases, Southern blot for larger size expansions. Pure Target creates an opportunity to consolidate more of that information into one targeted HiFi sequencing assay that can simplify the workflow and keep the results connected to the underlying molecule. For short-read users, repeat expansions are one of the clearest examples of where long-read sequencing can add information that short reads often struggle to provide. FMR1 is a good example of why sequence alone may not be enough. In Fragile X-related research, the CGG repeat size is important, but methylation context and allele structure are also important. The top panel shows methylation information, and the lower panel shows allele level repeat structure, including AGG interruption motifs. Pure target enrichment and HiFi sequencing can provide repeat structure, allele information, and methylation status together. That combination is valuable because it keeps multiple layers of information connected rather than forcing the lab to interpret disconnected measurements from separate assays. I want to close it with workflow readiness. For labs working in regulated or compliance-conscious environments, the sequence result is only part of the story. The electronic evidence trail also matters. Who accessed the system? What method was used? What changed? Who reviewed it? Who signed it? And how the record is retained. Vega includes capabilities that support 21 CFR Part 11 compliance readiness, including controlled access, audit trails, electronic signatures, and record retention. This does not replace labs' own validation responsibilities, but it can help reduce implementation burden and make the workflow easier to manage in a controlled environment. The take-home message is that Vega and SPRQ-Nx makes target HiFi sequencing more accessible, practical, and scalable. For current PacBio customers, this is an opportunity to extend HiFi into focused high-value targeted assays. For short-read customers, this is an opportunity to bring long-read HiFi sequencing to targets where short reads struggle most. Thank you.
Hi, and thank you for the introduction. I'd like to now take some minutes and talk about how Vega and SPRQ-Nx are great tools to help biopharma applications. As you probably know, the drug development process to make these life-changing medicines that we all need is really costly, long, and risky. So when we're starting from target identification and basic research, it takes about 10 to 15 years of research and development and clinical trials and costs about $2.6 billion to bring a drug to market. And only 5 to 8% of the drugs that start clinical trials will ever make it to market. So better sequencing data can really speed up and de-risk this process by providing comprehensive information to understand the drivers and regulators of disease, by identifying better candidates with desired properties and with more confidence and speed, by identifying potential safety concerns that may lead to clinical failure earlier in the process, and by potentially reducing time and cost to confident results. And PacBio highly accurate sequencing gives you confidence in your results. We provide powerful research tools for gene editing characterization, mRNA vaccine sequencing, AAV gene therapy, antibody development, and many more. And we'll go through each of these in a little bit more detail. The benefits of the Vega instrument with SPRQ-Nx chemistry for the drug discovery and development process are that you can sequence your way in-house. It gives you full control over the workflow and faster turnaround time than if you were sending it to an external service provider. It's also accessible, and the benchtop size and affordable pricing really can fit any budget and lab size. The SPRQ-Nx chemistry and the updates to our software now give you a path into regulated environments. Vega helps you now support 21 CFR Part 11 compliance to help you enable validation and compliance activities. And with this chemistry, you get more for less. The SPRQ-Nx chemistry provides higher output per sequencing run at lower input requirements and cost. So for gene editing research, what you want to do is to fully characterize your on and off target genome editing outcomes. And HiFi sequencing is really a great tool for that. Usually double-strand breaks that are created during gene editing, depending on the repair pathway that is being used, so either non-homologous end joining or homology-directed repair, can result in long and short indels. And it can also result in imprecise recombination, structural variants, concatenation of inserts, and other chromosomal instability. And HiFi is really one of the only methods that can characterize and assess all of these. So with HiFi reads, you can more comprehensively verify these gene editing techniques, and you can use amplification or amplification-free approaches, and then sequence with confidence to characterize your gene editing outcomes. This allows you to detect and accurately measure the efficiency of both on and off-target effects. It also allows to assess insertional mutagenesis and do insertion site analysis in greater detail, and overall helps you assess the potential safety of resulting constructs as guided by regulators. For mRNA vaccines, you can evaluate them for important criteria such as critical quality attributes, such as identity and the poly-A tail length. The sequencing the full-length mRNA at high accuracy helps you determine the size distribution and the sequence identity so that you can really confidently assess your products. It allows you to characterize the homopolymers, like poly-A, more accurately than with other sequencing methods, which helps you to optimize your construct and your process. And here's just an overview of how that works. So it's a little bit different than other regular RNA sequencing. You would add a GI anchor at the end of the poly-A tail, create a cDNA, and then essentially sequence this amplicon. And on the right-hand side, you see different poly-A tail lengths that have been characterized with this method, and you see that you can easily get up to about 100 A's in a row with HiFi sequencing. And for gene therapy research, I want to briefly just take a step back. AAV is a really great delivery vehicle in gene therapy, and usually people use up to 3 plasmids that they co-transfect into packaging cells, in which then AAV genome that consists a transgene flanked by 2 inverted terminal repeats is encapsulated into the viral capsid. And what you would expect is that all of these capsids contain the full-length recombinant AAV genome. But in reality, there's a distribution of empty, partial, and full capsids. And these are impurities, and there can also be non-AAV impurities that are packaged into this capsid. Measuring these impurities is really important because they can lead to toxicity and decrease the efficiency of the therapeutic. So with PacBio, you can really optimize your vector discovery and design so that you can really select for improved vector properties. Helps you improve your discovery and manufacturing to reduce the risk of failures. And you can easily implement end-to-end protocols to save you precious development time. On the bottom here, you see what a reference sequence might look like of the ITRs, the target construct transgene, with a plasmid backbone, and you see the variety of reads and the variety of genomes that could be packaged in HiFi really lets you distinguish all of them and really characterize how much of your capsids contain full-length genomes, versus with short reads, you get more ambiguous data. And last but not least, HiFi is also a great tool for antibody development. Short-read sequencing can't really cover the full length of several important antibody types, such as single-chain variable fragment or single-chain antigen binding fragment antibodies. And the Sanger sequencing that people often use suffers from low throughput, high hands-on time, and therefore limits the number of clones that can be analyzed. And with HiFi, you get really the full length, and you can do it all at the throughput that you want and need. So just to come back to the benefits of Vega and SPRQ-Nx for the drug discovery and development process, again, you can sequence your way in-house. These are now even more accessible long reads, giving you a path into regulated environments with this new instrument update, and you get more for less.
All right, so I'm going to be covering the microbial genomics applications and how Vega with SPRQ-Nx is helping to improve those. So Vega and SPRQ-Nx really brings increased value to microbial genomics applications, and is really through these 4 main pillars. So the first one being faster Vega runtimes enables 24-hour turnaround time from sample to reports. The next one is lower input requirement per Vega Smart Cell, really allows access to more sample types that may be lower in biomass. The other one is higher yield and lower cost per Vega run, really increases cost effectiveness across multiple microbial applications. And lastly, 21 CFR Part 11 compliance enablement on Vega really add value in more regulated environments, being able to add Vega to those labs that are interested in long read sequencing. So we have multiple solutions for microbiology applications, and these through a variety of different library prep options. And so first is the full-length amplicon sequencing of full genes. So this can be 16S for bacteria, it can be ITS for fungal, 18S for eukaryotes. You can also do entire operons if you'd like. And the value this gives is more classified reads, and importantly, at the species and strain level in comparison to what you would typically get with short read partial based amplicon sequencing. And so the library prep solutions we have for this are first Connects, which concatenates multiple amplicons together to take full advantage of the HiFi long reads. The other thing we have is a partnership with Zymo Research, where they have a quick 16S full-length library prep kit, which gives you all the PCR reagents up front in a nice plate kit format. And then N6 has an Icon PCR machine, which really allows for auto-normalization and single well amplification. And then into the whole genome sequencing application. So we have microbial isolate, WGS, which really gives, with HiFi, you get closed complete chromosomes and plasmids, really reference quality genomes, and there's no need to do a hybrid-based assembly approach with this. And you also get native methylation calling with it, as long as you don't use an amplification-based approach. The next is shotgun metagenomics. Through this, we have metagenome profiling, which is read-based mapping to reference databases. With HiFi, you get really high precision and recall for taxonomy classifications. And importantly, you get more and richer functional annotations than you would with short reads. And then lastly, metagenome assembly. We were able to get many high-quality MAGs, or metagenome assembled genomes, many of which are actually single contig, complete or near complete MAGs, and of which are many of them are actually at the strain level. This is also without needing a hybrid-based approach with polishing with short reads. And for this, we have lots of different library prep solutions, our HiFi Plex Prep, our HiFi Prep, SmartBell Prep Kit, as well as the Amplify protocol, which is for ultra-low input sample types. And then lastly, Sequel, which is another one of our partners, with their long-plex multiplexing kit, which allows for lower cost per sample, as well as high multiplex and easier workflow capabilities. So now I'm going to walk through those different value adds for SPRQ-Nx on Vega for these different applications that they add value for. So the first is the faster Vega runtimes with SPRQ-Nx. Really enables less than 24-hour turnaround time from raw sample to report. So the primary applications of interest for this is really full-length amplicons. So again, this can be bacterial 16S, eukaryotic 18S, fungal ITS. And you can see that the relative sizes of those are 1.5 kb or 800 base pairs. So you can run them at either 4-hour runs for these first two, or a 2-hour run for a fungal ITS. And so I've kind of mapped out the typical workflow for this, starting at DNA and isolation and going all the way through analysis and report generation. You can see that the typical turnaround time for this from sample to report is as little as 10 hours, which is really incredible. You can do this within a single day. The other addition of SPRQ-Nx is allowing on Vega is the lower input requirement. So this is allowing access to more sample types, for example, like water, which can be low biomass. So here's an example paper that came out last year on wastewater in particular. This is from Rob Light's lab at UCSD, and this is where they were surveilling the campus there for wastewater surveillance. So with PacBio HiFi, they're able to unlock species-resolved antimicrobial resistance surveillance in these wastewater systems. They're able to, with the HiFi long reads, they're actually able to place the antimicrobial resistance genes directly in the species and strain context, which is really beyond what you can do with short reads. They're also able to find and reveal AMR dark matter in unclassified and emerging microbes. They're also able to identify high-risk multi-drug resistant taxa, allowing for detecting species that were carrying from 10 to as many as greater than 30 unique antimicrobial resistance genes. And lastly, they're able to capture plasmid-associated resistance. So this improved the detection of those mobile antimicrobial resistance genes, which are really critical for tracking horizontal gene transfer. The next advantage of SPRQ-Nx on Vega is it has a higher yield and a lower cost, which really increases the cost effectiveness of multiple applications. And so the specific applications that get a boost with SPRQ-Nx on Vega is microbial isolate WGS, full-length amplicons, again, could be 16S, ITS, or whatever, using both Kinnex or the standard approach. And in particular, the one that gets the most boost is shotgun metagenomics, both profiling and assembly. And this is because it requires more depth and coverage than the other sample types or the other applications. So an example here on the right is of HiFi deep sequencing for metagenomics of 1 terabase. And so you can think of this as like sequencing a soil sample very deeply or a marine water, or you can even think about it as multiplexing multiple human gut samples, for example. And so you can see back with the Sequel 2, this would take lots of acquisitions of SMRT cells, many days, 63 days, as well as a lot of money. So this is $63,000 to get 1 terabase of metagenome data. The Vega version 1 already helps a lot with this. It brings it down to 17 days, $19,000. But now with SPRQ-Nx, it brings it further, faster, with 13 days of sequencing and only $13,000 USD, which is really getting close now to what it is with SPRQ-Nx on the Revio platform, but it's a lot more accessible as far as the capital purchase price. And so when looking at the differences between the Vega system version 1 and the Vega system with SPRQ-Nx, this is showing the microbial genomics applications, the recommended maximum number of samples per SMRT cell, the estimated yield per sample, and the cost estimate per sample. And so Vega is on the left, SPRQ-Nx is on the right. I have highlighted or in magenta color the differences between the two, the added benefits. So you can see you get more coverage for microbial isolate whole genome sequencing with SPRQ-Nx. For metagenomics, for both profiling and assembly, you're able to multiplex more samples for both of those. In addition, they are cheaper per sample for any of the library prep options that we have. So the HiFi Plex, the HiFi Prep, the Amplify protocol, or the Sequel Longplex. And then for 16S or any amplicon, you either get more reads with the standard-based approach per sample, or you can multiplex more samples with the Kinnex 16S. And then we get a lot of, when having conversations with folks in the field, we get a lot of concerns about multiplex levels and being able to fill a cell with 384 microbial isolate whole genomes, for example, which is totally understandable. Not a lot of labs are super high throughput like this, especially with the Vega system. So what this is showing is it's still very cost effective, even as low as like a 24-plex. So you can see 24 samples on a single Vega SPRQ-Nx cell. It's still only about $70. If you were to go up to a 96-plex, you can see a significant savings per sample at 33, which is very close to what it is at 384. And then lastly, the 21 CFR Part 11 compliance enablement on the Vega adds value in more regulated environments because it makes it easier to implement Vega in these regulated labs that are currently interested in adopting long-read sequencing. And so an example of a lab like this is Intus Biosciences Gut ID, which is a at-home microbiome test kit that you can order and collect a sample, and then they will sequence and give the analysis results for you at home. And that's it for me. Thanks for your time. Happy to take any questions in the Q&A portion.
Hi, everybody. Thank you for your attention during our recordings. We're going to go ahead and jump into the Q&A. We have some questions that came in during the presentation where I will just read off questions and either answer or call on one of our panelists to help answer. The first question is, will there also be multi-use SMRT cells for the Vega system? While we will be using SPRQ-Nx chemistry on the Vega system, which is identical to that on the Revio system, the Vega system will not have the SMRT cell multi-use capabilities at this time. We are evaluating bringing that feature to the Vega system at a future date. Next question. Does the Vega SMRT cell have the same number of ZMWs than that of the Revio? And do you foresee the Vega system deploying a multi-use capability in the future? So I just answered the multi-use one. For the yield, essentially, relative to the Revio system, The Revio and Vega systems are both integrated systems, with each system engineered essentially to deliver an optimal and consistent result. With the SPRQ-Nx update on the Vega system, the system and all of the workflows have been optimized along with the improved SPRQ-Nx chemistry formulation to deliver increased yields on the Vega system up to 90 gigs HiFi per run. So the systems are similar, but they have been engineered and optimized to deliver different yields based off of different needs. Next question. Can 2-hour and 4-hour runs also be used for sequencing linearized plasmid DNA with a size of 6 kb? We do not need millions of reads, as it should be used to release testing of plasmid DNA. Nadia, could you take a swing at this one?
Of course, happy to. So the shorter runtimes are a trade-off with accuracy that also depend on the length of the molecule. So since you say that your length is about 6 kb, I think you might be sacrificing too much quality if you do a 2 or 4 hour runtime. I might suggest to go for a 12-hour runtime at least, if not 24, since you're using it as a release test. You'll get sequencing results from 2 or 4 hour runtimes. You just have lower quality scores for that. And with the millions of reads, you will absolutely get millions of reads from Vega SMRT cell run. How many reads per plasmid you get is largely a function of your level of multiplexing. So depending on how many plasmids or how many samples you multiplex on the SMRT cell is going to determine how many reads exactly you get per sample.
Thanks, Nadia. Next question. I think I'm also you're interested in sequencing plasmid DNA, circular and linearized, with and without poly A tails up to 140 base pair. Will there be a workflow for the analysis of data generated by sequencing these sample types on the Vega platform? We would need it for release testing of GMP plasmids. Additionally, can the poly A tail length be accurately determined on the plasmid DNA?
Yeah, thanks. Happy to take this one. So I'll start with the poly A length on the plasmid level. You can absolutely do that. And it's actually a bit easier than if you're doing it from the mRNA, since you don't have to create, this construct first that converts the RNA into cDNA, etc. So poly A tail on linearized and circular plasmids works really well, and we have other customers doing that as well. In terms of will there be a workflow for the analysis of this data, the short answer is you'll just run it like regular HiFi sequencing. For the bioinformatics piece, we don't currently have a PacBio developed workflow. Our partner Sequel has put together a pipeline that you can find in our plasmid sequencing app brief.
Great. Thank you, Nadia. Next question is, can you clarify, SPRQ is a new version of sequencing reagents, SMRT cells, or both. The update for Vega, for the SPRQ-Nx chemistry release, there is a new Vega sequencing plate, a Vega SPRQ-Nx sequencing plate, and then there is also a new Vega SPRQ polymerase kit. So the two products that are being updated with this release are the polymerase kit, which is used to prepare the library for sequencing on Vega, and then the actual sequencing plate that you put onto the Vega system that contains the sequencing reagents. The SMRT cell for Vega, because it is not getting that multi-use function like we released on Revio, will be the same as the on market today. So no change with SMRT cell. Next question. Are there scenarios where non-SPRQ chemistry or non-SPRQ-Nx chemistry is still preferable, or would that be phased out? There are really no benefits of running the current on-market Vega chemistry versus the SPRQ-Nx chemistry. SPRQ-Nx delivers, like you heard in the presentations, increased performance, increased yields, decreased DNA input requirements. With the software, we add additional methylation detection. So there are really only, and it's cheaper actually than the V1 chemistry. by about 10% list price. So there are only really benefits to switching to SPRQ-Nx chemistry on Vega. We will look to at the adoption rate of SPRQ-Nx and determine what to do with the current chemistry on Vega. But I am anticipating a broad and fast adoption of SPRQ-Nx on Vega due to the benefits. So it is likely that we end of life that current chemistry sometime in the foreseeable future. But it will be commercially available after launch until we do that end of life. Next question of whether are the sequencing reagents for 2 and 4 hour runs different or the same for 24-hour runs? It's all the same. 2 4-hour, 12-hour, 24-hour runs, they all use the same chemistry and the same sequencing plate. So you order one sequencing plate and can use it with any movie time. Next question is, when making use of the SPRQ-Nx chemistry on Vega, what happens when you sequence a library with a large distribution, about 5 to 10 kb? Do you get full-length reads, but less of them overall, or will the system show preference for the short reads? So this is highly dependent on the movie length. If you choose for example, 4-hour movie time with a library of 5 to 10 kb in size, you won't generate many HiFi reads simply because the movie wasn't long enough. But if you run a 24-hour movie, you'll essentially max out the potential HiFi read output due to the movie length. So it's not necessarily a function of the system, but it's more a function of the movie length relative to the library size. Next question. Can you comment on how SPRQ-Nx on Vega actually increases yields? Primarily a computational improvement. Read lengths also improve. Do we need to prep libraries to be longer? And how does this impact base calling time? So I'll start with the libraries. You don't have to prep libraries to be longer. You can run the exact same libraries that you've been running previously. If you would like to go longer, you can always do that, but you don't have to because of SPRQ-Nx chemistry. And how are we increasing yields? It's primarily due to increase in polymerase read length, which just increases HiFi conversion and provides more robust yields. And how does that impact base calling time? There is no impact. Again, the system has been optimized for this chemistry, so your post-processing time is roughly the same with SPRQ-Nx than it was with the current chemistry. Next question is Vega versus Vega SPRQ-Nx. Is there any change for library prep or only the different kits? There's no change for library prep. Library prep is the exact same as it is today. And it's also the same as the Revio. So if you make a library for the Revio, it can also be run on Vega and vice versa. The only difference here is the new kits and some volume changes in the workflow to support the lower DNA input requirements. Next question is, how does the system compare against the nanopore systems? There are many nanopore systems to compare the Vega system to, but what I'll say, how the Vega system differentiates itself from any nanopore system is 1, the simple workflow and the accessibility, the ease of use, and then 2, the reliability and consistency. Vega runs are extremely reliable, extremely consistent, and even with the increased yields that we get with SPRQ-Nx chemistry, we're still seeing that consistency. So when users and customers run on the Vega, they know what they're going to get. And I think that's a little bit different than when you run on ONT. The DNA methylation detection accuracy, that was a question of how it compares to current performance. We have a new 5-HMC caller, which we didn't have previously, so that's net new performance. The 5MC and 6MA calling that gets done on instrument on the Vega system with every run has been slightly improved relative to the previous release or previous version or the current on-market version of Vega. So you'll see a little bit of improvements there in methylation calling. Let's see, next question. are there plans to expand your pure target carrier panel for other difficult regions, for example, PKD1 or pseudogenes? Gianluca, if you are still on the call, I think this would be a great question for you.
Yes, that's a great question. However, customers have the opportunity to develop their own custom target panels for pure target and add those targets to existing carrier panels that we have.
Great, thank you. And then, as of right now, our last question is, the input quantity of DNA will be 500 nanograms, will the DNA input quantity be 500 nanograms with the long plex from SeqWell also? Let's see, Nadia or Jeremy, would you be able to answer that?
Yeah, so just a quick reminder, the 500 nanograms is what's needed for input for a single SMRT cell run. So if you're multiplexing, it can be less per sample. For SeqWell Longplex, their recommendation for PCR-free, I believe, is 250 nanograms per sample, and for PCR plus is 150 nanograms per sample.
Great. And that was our last question. So thank you all for attending. We appreciate your time and attention. Be on the lookout again for follow-up emails with links to recordings, literature, etc. And again, just appreciate your time. Thank you, everybody.